practically gene editing with CRISPR-Cas9 experiment. Imagine holding the power to rewrite the very blueprint of life. What once seemed like science fiction is now a reality thanks to CRISPR-Cas9, a groundbreaking gene editing tool. But how does it actually work? And can we truly alter DNA with precision? Today, we're diving into the fascinating world of CRISPR with a hands-on experiment that will show you exactly how scientists are editing genes in real time. Whether you're a biotech enthusiast, one introduction to CRISPR-Cas9, how it works, and its applications. CRISPR-Cas9 is one of the most groundbreaking technologies in modern biotechnology, revolutionizing the way we approach genetic engineering. But what exactly is CRISPR, and how does it work? At its core, CRISPR, clustered regularly interspaced short palindromic repeats, is a defense mechanism originally discovered in bacteria. It allows them to recognize and cut viral DNA, preventing infections. Scientists realized that this natural system could be repurposed as a precise and programmable tool for gene editing. By pairing CRISPR with the Cas9 enzyme, we can now target specific DNA sequences and modify them with unparalleled accuracy. How CRISPR-Cas9 works. Guide RNA, gRNA selection. Scientists design a short RNA sequence that matches the DNA region they want to edit. This guide RNA acts as a homing device, leading the Cas9 enzyme to the exact location in the genome. Cas9 enzyme activation. Once at the target site, Cas9 acts like molecular scissors, cutting both strands of the DNA. DNA repair and modification. After the cut, the cell attempts to repair the DNA. Scientists can use this process to introduce new genetic material, remove unwanted mutations, or disrupt specific genes. This precise editing capability makes CRISPR-Cas9 an incredibly powerful tool with applications that span multiple fields. Applications of CRISPR-Cas9 medicine and disease treatment. CRISPR is being explored as a potential cure for genetic disorders like sickle cell anemia, cystic fibrosis, and even certain types of cancer. Scientists are developing gene therapies that could correct mutations at their source, offering hope for millions of patients. Agriculture and food security. By modifying plant DNA, CRISPR allows scientists to create crops that are more resistant to pests, droughts, and diseases. This can improve food production efficiency and reduce the need for harmful pesticides. Synthetic biology and biomanufacturing. Researchers are using CRISPR to engineer bacteria and yeast to produce biofuels, pharmaceuticals, and even lab-grown meat. This could lead to more sustainable industrial processes. Ecosystem and conservation. CRISPR is being considered for applications like reviving extinct species, de-extinction, and controlling invasive species. Scientists are also exploring ways to use gene editing to combat vector-borne diseases like malaria by modifying mosquito populations. The future of CRISPR. While CRISPR-Cas9 is a revolutionary tool, it's not without challenges. Ethical concerns, potential unintended mutations, off-target effects, and regulatory hurdles must be carefully addressed as the technology advances. However, as research progresses, CRISPR is expected to unlock new frontiers in genetics, medicine, and beyond. In this video, we'll take you through a practical CRISPR-Cas9 experiment to demonstrate how scientists edit DNA in real life. Let's get started. 2. Preparing for the CRISPR-Cas9 experiment, tools and materials you need. Before diving into the exciting world of gene editing, it's crucial to understand the tools and materials required for a CRISPR-Cas9 experiment. Whether you're working in a high-tech laboratory or exploring this concept in a DIY biohacker space, the right resources are essential for ensuring the experiment's success. Let's break down what you'll need to get started. CRISPR Guide RNA. gRNA. The guide RNA, gRNA, is one of the most important components in the CRISPR-Cas9 system. It acts like a GPS system, directing the Cas9 enzyme to the exact location in the DNA sequence that you want to edit. The gRNA is designed to match the target gene, ensuring precision. How it's used. The gRNA is synthesized to complement the DNA sequence you aim to edit. It's crucial that the sequence is highly specific to avoid off-target effects, which could lead to unintended changes in other parts of the genome. Why it matters. The effectiveness of your CRISPR experiment hinges on the accuracy of this guide. A single mismatch can result in failed edits or worse, unpredictable consequences in the genome. Cas9 enzyme. The Cas9 enzyme is the molecular scissors that do the heavy lifting in gene editing. Once the gRNA guides it to the target location, Cas9 makes a precise cut in the DNA. This cut is crucial for initiating the process of gene modification. How it's used. Cas9 can be introduced into the cells either by plasmids, circular DNA molecules, or through a direct injection of the enzyme itself. Once inside the cell, it locates the target sequence and cuts the DNA. Why it matters. Cas9's precision is vital for ensuring the edit is made exactly where you intend. Any error in the cutting process can lead to a failure in gene modification or cause undesirable mutations. DNA sample, target DNA. The target DNA is the genetic material you want to modify. This could be a segment of DNA from any organism, human, plant, or animal. In most experiments, scientists work with cells from model organisms like bacteria or yeast before moving to more complex systems like plants or mammals. How it's used? You'll need to isolate the DNA from your organism and identify the specific sequence to edit. This is where the precision of your gRNA and Cas9 becomes essential. Why it matters. The quality of the DNA sample directly impacts the efficiency of the gene editing process. Contaminated or degraded DNA will lead to poor results. Repair template, optional. In some experiments, you'll need a repair template, a piece of DNA that serves as a guide for repairing the DNA cut made by Cas9. If you want to insert new genes or correct mutations, the repair template is crucial for providing the necessary genetic material. How it's used. The repair template is designed to match the DNA sequence at the site of the cut. After Cas9 makes the cut, the cell will use the repair template to insert or replace the genetic material. Why it matters. The accuracy of this repair is essential for successful gene modification. It ensures that the desired genetic change is introduced without disrupting other parts of the genome. Delivery method, electroporation or viral vectors. To introduce the CRISPR components, gRNA, Cas9, and repair template into the target cells, you'll need a reliable delivery method. Two common approaches are electroporation and viral vectors. Electroporation. This technique uses an electric field to create temporary pores in the cell membrane, allowing the CRISPR components to enter the cell. Viral vectors. Viruses are engineered to carry the CRISPR components into the target cells, taking advantage of the virus's natural ability to infect cells. Why it matters. The delivery method you choose will determine how efficiently the CRISPR components reach the target cells. Poor delivery could lead to low efficiency or failed edits. Laboratory equipment. To carry out the experiment, you'll also need basic lab equipment like pipettes, test tubes, petri dishes, centrifuges, and growth media to culture the cells. The cleanliness and sterility of the lab environment are also crucial for preventing contamination, which could affect your results. Why it matters. Proper equipment ensures that each step of the experiment is performed accurately and safely. Contamination can lead to incorrect data or even unsafe gene modifications. Monitoring and analysis tools. After you've completed the gene editing process, it's essential to verify the success of the edit. This can be done through DNA sequencing and PCR analysis, which allow you to confirm that the target DNA has been successfully modified. Why it matters. Verifying the results is crucial to ensure that your experiment has been successful. DNA sequencing will tell you if the edits are as expected or if there have been off-target effects. With all the right tools and materials in place, you're now ready to perform your CRISPR-Cas9 gene editing experiment. The precision, control, and possibilities offered by CRISPR-Cas9 are what make it a revolutionary technology in the field of genetics. However, it's important to note that this technology is complex, requiring careful attention to detail at every step of the process. In the next part of our video, we'll walk you through the step-by-step procedure of actually conducting a CRISPR-Cas9 experiment. Stay tuned. 3. Performing the CRISPR-Cas9 gene editing experiment, step-by-step process. Now that we've prepared all the necessary tools and materials, it's time to dive into the actual gene editing process using CRISPR-Cas9. In this section, we'll take you through each step, from designing the guide RNA to editing the DNA and confirming the results. Let's break down the steps clearly so you can see exactly how gene editing happens in real time. Step 1. Designing the guide RNA, gRNA. The first step in any CRISPR-Cas9 experiment is to design the guide RNA. The gRNA is responsible for guiding the Cas9 enzyme to a specific location in the DNA where the cut will be made. To do this, you'll need to identify the target sequence in the genome you want to edit. This could be a gene that you want to deactivate, modify, or replace. How it works. The gRNA sequence is designed to be complementary to the target DNA sequence. It must bind to the exact region where the edit needs to occur. Tools like CRISPR design software can help predict the most efficient gRNA sequences based on your target gene. Why it matters. The success of your experiment depends heavily on how accurately the gRNA binds to the target. A poorly designed gRNA might lead to off-target effects or failed edits. Step 2. Preparing the Cas9 enzyme and gRNA. Once your gRNA is designed, the next step is to prepare the Cas9 enzyme and combine it with the gRNA. The Cas9 enzyme will be guided by the gRNA to make a precise cut in A at the target site. How it works. Cas9 is mixed with the gRNA to form a complex that can be delivered into the target cells. The Cas9 enzyme acts as molecular scissors, cutting the double-stranded DNA at a specific location. Why it matters. The efficiency of the DNA cutting is directly tied to the quality of the Cas9 enzyme and the gRNA. If they are not combined correctly, or if the delivery method is not efficient, the cut may not occur as intended. Step 3. Delivery of CRISPR components into target cells. The next step is to introduce the CRISPR components, Cas9 and gRNA, into the target cells. This can be done in several ways, depending on the type of organism and the method of experimentation. Two common techniques are electroporation and viral vectors. How it works. Electroporation uses an electric field to create temporary pores in the cell membrane, allowing the CRISPR components to enter the cell. Viral vectors use modified viruses to deliver the CRISPR components directly into the cells. Why it matters. The method you use to introduce the CRISPR components will affect how efficiently the DNA is edited. Poor delivery can result in low transformation rates or failed experiments. Step 4. Gene editing. Cas9 makes the cut. After the CRISPR components enter the cells, Cas9 gets to work, making a precise cut in the target DNA. The guide RNA directs Cas9 to the correct sequence, where the enzyme makes a double-strand break in the DNA. How it works. Once Cas9 cuts the DNA, the cell's natural repair mechanisms kick in. There are two main pathways for repair: non-homologous end joining (NHEJ) and homology-directed repair (HDR). NHA often leads to insertions or deletions, indels at the cut site, potentially disrupting a gene. HDR can be used to insert new genetic material by providing a repair template. Why it matters. The choice of repair pathway determines the outcome of the gene edit. If you want to insert a new gene or correct a mutation, HDR is often preferred. If you want to deactivate a gene, NA is typically used. Step 5. Monitoring the cells after editing. Once the DNA has been edited, the next step is to allow the cells to recover and divide. Over the next few days, you'll want to monitor the cells to see how they respond to the gene edit. How it works. Cells that have successfully incorporated the edit will continue to grow and divide, while cells that failed to incorporate the changes will either die or fail to propagate. Why it matters? It's crucial to monitor the cells to assess the efficiency of your gene editing process. This step helps identify which cells have been successfully edited and which haven't, allowing you to focus on the ones with the desired change. Step 6. Verifying the gene edit. Once the cells have been edited and grown, it's time to verify that the gene modification was successful. This is typically done using DNA sequencing or polymerase chain reaction, PCR. How it works. PCR can amplify the edited region of the DNA, making it easier to confirm whether the edit has occurred. DNA sequencing provides a detailed readout of the DNA sequence, allowing you to compare the original sequence with the edited one to confirm the exact changes. Why it matters. Verifying the gene edit is essential for confirming the success of the experiment. This ensures that the desired genetic modification has occurred and that no unintended changes have been made. After completing these steps, you'll have successfully performed a CRISPR-Cas9 gene editing experiment. From designing the gRNA to confirming the results, each step is crucial for achieving precise and accurate genetic modifications. While this technology offers incredible potential for research, medicine, and biotechnology, it also requires careful attention to detail and expertise. As CRISPR technology continues to evolve, we can expect even more exciting advancements and applications in the near future. In the next section of this video, we'll discuss the challenges and ethical considerations that come with gene editing. Stay tuned. 4. Observing the results and analyzing the CRISPR-Cas9 gene edit. Once you've completed the gene editing process, the next critical step is to observe the results and analyze the effectiveness of your CRISPR-Cas9 experiment. At this stage, you'll need to confirm that the gene modification has occurred as planned and assess whether the edited cells exhibit the desired traits or characteristics. Let's dive into the process of observing the results and analyzing the data. Step 1. Observing the edited cells. After the CRISPR components, Cas9 and guide RNA, have been delivered into the cells and the DNA cut has been made, the edited cells need time to recover and divide. During this recovery period, it's essential to monitor the cells closely to track their growth and behavior. How it works. Depending on your experimental design, you may observe specific markers in the edited cells. For example, if you've introduced a fluorescent protein or a selectable marker into the gene, you'll be able to visually track whether the cells have successfully taken up the genetic modification. Why it matters. This stage allows you to confirm whether the cells have survived the editing process and whether they are expressing the modified genes. If the cells don't exhibit the expected changes or show signs of stress or abnormal growth, it could indicate that the editing was unsuccessful. Step 2. Using PCR to amplify the target region. Once you've observed the cells, it's time to verify whether the specific gene modification has occurred. One of the most commonly used methods to analyze gene edits is polymerase chain reaction, PCR. PCR is used to amplify specific regions of DNA, making it easier to detect and analyze the changes at the target site. How it works. After extracting the DNA from the edited cells, you use PCR to amplify the region of interest, the part of the genome that you targeted with CRISPR. If the gene has been successfully edited, the amplified DNA will show the specific mutations, insertions, or deletions you intended to introduce. Why it matters. PCR helps confirm that the desired change has been made at the molecular level. By comparing the amplified sequence to the original sequence, you can determine whether the gene was edited correctly and whether the modification is consistent across different cell populations. Step 3. DNA sequencing for detailed verification. While PCR provides a quick and reliable way to detect gene edits, DNA sequencing is the gold standard for verifying the exact changes made in the genome. Sequencing provides a detailed readout of the DNA, allowing you to compare the original and edited sequences to identify any discrepancies or unintended changes. How it works. Once PCR has amplified the target region, you can sequence the DNA to get a complete, high-resolution picture of the gene edit. The sequencing process reads the nucleotide sequence of the DNA, providing a clear and precise view of what changes have been made. Why it matters. DNA sequencing allows for the most accurate verification of the gene edit. It ensures that the modification is exactly as intended and helps you identify whether there are any off-target effects, unintended edits at other locations in the genome. This is a crucial step in confirming that the CRISPR-Cas9 system has worked precisely and without errors. Step 4. Assessing the phenotypic changes, if applicable. In some experiments, particularly those involving organisms like plants or animals, the goal of gene editing is to produce observable phenotypic changes. This might include altering a specific trait, such as disease resistance in plants, or modifying physical characteristics in animals. How it works. If your experiment involves studying a phenotypic change, you'll need to monitor the organisms over time to see how they express the genetic modification. For example, if you've edited a gene related to color in a plant, you would observe whether the plants show the desired color change. Similarly, in animals, you might look for changes in behavior, growth, or health. Why it matters. Phenotypic analysis allows you to correlate the genetic modification with observable traits, helping to validate the effectiveness of your edit. It also provides a way to understand the broader impact of gene editing on the organism, which is especially important in agricultural or biomedical research. Step 5. Evaluating off-target effects. One of the most significant concerns in gene editing is the potential for off-target effects, where the CRISPR system makes unintended edits to other parts of the genome. These unintended mutations can lead to harmful consequences, especially in medical applications. How it works. To check for off-target effects, you can use high-throughput sequencing technologies, such as whole genome sequencing or deep sequencing, to examine the entire genome for unintended edits. By comparing the pre-edit and post-edit sequences across the entire genome, you can detect any off-target changes. Why it matters. Ensuring that the gene editing process is precise is critical, especially when applying CRISPR in medical or clinical settings. Detecting and minimizing off-target effects is a major part of ensuring the safety and efficacy of gene editing. Step 6. Final confirmation and documentation. After confirming that the gene edits have been successfully made and that the results meet your expectations, it's time to document and analyze the final outcomes. You'll want to record your findings and keep track of the specific changes made, the methods used, and the results observed. How it works. Data from PCR, sequencing, and phenotypic analysis should be compiled into a comprehensive report. This includes an assessment of whether the gene editing was successful, how the cells or organisms have responded, and whether any unexpected effects were observed. Why it matters. Proper documentation of results ensures that your experiment can be replicated and validated by others in the field. It also provides a clear record of the outcomes, which is essential for future research and development. Observing and analyzing the results of a CRISPR-Cas9 gene editing experiment is a critical part of the process. From confirming the success of the edit using PCR and DNA sequencing to assessing any phenotypic changes and checking for off-target effects, each step provides valuable insights into the accuracy and effectiveness of your gene modification. With careful analysis, you can ensure that your experiment has achieved the desired outcome and that the technology is being used safely and responsibly. As CRISPR-Cas9 continues to evolve, we can expect even more precise and efficient methods for editing genes, making it a game changer in fields like medicine, agriculture, and biotechnology. Stay tuned for more exciting developments in this groundbreaking field. 5. Challenges and future potential of CRISPR technology. CRISPR-Cas9 has revolutionized the world of genetic engineering, opening up vast possibilities for gene editing. From medical breakthroughs to agricultural innovations, CRISPR promises to change the way we approach genetic diseases, crop improvement, and more. However, as with any groundbreaking technology, CRISPR comes with its own set of challenges. At the same time, its future potential is boundless, and overcoming these obstacles could lead to a new era of genetic science. Challenges facing CRISPR technology. Off-target effects. One of the biggest concerns with CRISPR-Cas9 is off-target effects, unintended modifications to parts of the genome that were not meant to be altered. While CRISPR has been praised for its precision, it's not always perfect. If the Cas9 enzyme makes a cut at an unintended location, it could lead to harmful mutations, which might cause unintended diseases or complications, especially in human therapeutics. Current solutions. Scientists are working on developing more accurate and specific versions of the Cas9 enzyme to reduce off-target effects. Modified versions like high-fidelity Cas9 and CPF1, a different Cas protein, offer better precision. Additionally, new delivery methods are being explored to improve the targeting accuracy. Ethical and safety concerns. Gene editing, especially in humans, raises serious ethical and safety questions. Editing the human germline, the genetic material passed on to offspring, could have long-lasting effects that extend beyond the individual, potentially altering the human gene pool. There are concerns about unintended consequences, such as introducing new diseases or genetic mutations that may not manifest until future generations. Current solutions. Many countries have strict regulations around human germline editing, focusing on somatic, non-reproductive cell editing for therapeutic purposes. Ethical discussions are ongoing, and an international consensus on the responsible use of CRISPR is critical for its future applications. Delivery challenges. Efficiently delivering the CRISPR-Cas9 components, specifically the Cas9 protein and guide RNA, into the target cells is still a challenge. The delivery methods need to be highly efficient and non-toxic. Methods like electroporation and viral vectors work, but they have limitations in terms of efficiency, cost, and safety. Current solutions. Researchers are experimenting with new delivery vehicles, such as nanoparticles, which could improve the delivery efficiency of CRISPR components. Advances in nanotechnology and biomaterials are expected to help overcome current delivery barriers, making CRISPR more accessible and effective. Regulatory and legal barriers. The rapid development of CRISPR technology has outpaced the legal and regulatory frameworks that govern genetic editing. Laws related to gene editing vary from country to country, creating a complex environment for researchers and companies in the field. Current solutions. Governments and regulatory bodies are increasingly focusing on creating clearer guidelines for the use of CRISPR technology, particularly in the realms of medicine and agriculture. International collaboration will be crucial to ensure that CRISPR is used ethically and safely across borders. Future potential of CRISPR revolutionizing medicine. CRISPR holds tremendous potential for curing genetic diseases and advancing personalized medicine. By targeting the root causes of diseases like sickle cell anemia, cystic fibrosis, and muscular dystrophy, CRISPR offers the possibility of one-time, life-changing treatments. Future developments. As our understanding of CRISPR improves, it's expected that gene editing will become more widespread in clinical settings, potentially treating a wide range of genetic disorders. Personalized medicine, where treatments are tailored to an individual's genetic makeup, could become commonplace, revolutionizing how we approach healthcare. Advancements in agriculture. In agriculture, CRISPR is already being used to create genetically modified crops that are more resistant to disease, drought, and pests. This could significantly increase food production, which is crucial as the global population continues to grow. CRISPR also holds promise for improving the nutritional content of crops, potentially addressing food security issues. Future developments. As CRISPR technology becomes more refined, we could see crops with enhanced traits such as higher yields, better resistance to climate change, and improved nutritional value. This would not only benefit farmers, but also provide healthier food options for consumers worldwide. Environmental impact. Gene editing could play a major role in biodiversity conservation and ecosystem management. For instance, CRISPR could be used to help protect endangered species by introducing genes that make them more resilient to disease, climate change, or habitat loss. Additionally, CRISPR could be used to control invasive species that threaten ecosystems. Future developments. One exciting possibility is the use of CRISPR in gene drives, which can spread a genetic modification through a population rapidly. This could be used to eradicate diseases like malaria by genetically modifying mosquitoes to be resistant to the disease, potentially saving millions of lives. Bioengineering and synthetic biology. As we move toward a future where synthetic biology and bioengineering become more prevalent, CRISPR will play a crucial role in designing organisms with entirely new capabilities. Whether it's creating microorganisms that can produce renewable energy, engineering bacteria to break down pollutants, or designing synthetic life forms for space exploration, CRISPR's precision will make it an invaluable tool. Future developments. The future of CRISPR lies in its ability to create entirely new biological systems, which could lead to breakthroughs in fields like biofuels, environmental remediation, and even space exploration. Scientists are just beginning to scratch the surface of CRISPR's potential in bioengineering. While CRISPR-Cas9 faces significant challenges ranging from off-target effects and ethical concerns to delivery difficulties and regulatory hurdles, the potential benefits of this technology are immense. The ability to edit genes with precision opens up incredible possibilities in medicine, agriculture, environmental conservation, and beyond. As research continues and technology improves, CRISPR could pave the way for a future where genetic diseases are cured, food security is improved, and biodiversity is preserved. However, careful attention to its ethical, legal, and safety implications will be essential for ensuring that CRISPR is used responsibly and effectively. As we look toward the future, The possibilities for CRISPR are limitless, and overcoming these challenges could lead to one of the most significant breakthroughs in modern science. Stay tuned for what's next in this incredible journey. As we conclude our exploration of CRISPR-Cas9, the future of gene editing is in our hands. If you're as excited as we are about the possibilities of this groundbreaking technology, don't forget to like, subscribe, and hit the notification bell for more updates on CRISPR and other innovative technologies. Join the conversation by sharing your thoughts in the comments below. How do you think CRISPR will shape our future? Stay connected and be part of the revolution in genetic science. Let's unlock the potential of tomorrow, today.